Hello, class. So yesterday we left off with understanding some of these basic ideas of photosynthesis and how everything happens. So today we're going to get into the details. A lot of this I don't need you to know, but I want to expose you to it because if any of you would like to go on and do something more biology related, like maybe a doctor or a scientist, you're going to have to learn photosynthesis in detail again. So if I can just expose you to it now, and also if you want to learn it at a higher level than what we have so far, here is the video for you. If you don't want to, I still want you guys to watch this. So sorry you're stuck, even if you don't want to, but I think it is really important and helps us understand exactly where everything is going when we understand the detailed steps. So the first step of photosynthesis is our light-dependent reactions. So I don't think I mentioned this in our first one, but just to be clear, dependent means that we need it, right? So our light dependent is a process that has to have light, whereas our light independent is not. Sometimes in the United States, even a child is called a dependent of a parent, as if the child needs the parent to live for shelter, food, money, and whatever. Anyways, back to our steps which are in our light dependent process. So the first thing, step one, is our photosystem 2. Again, they were named by the way they were discovered, not the way that they are in order. So photosystem 2 comes before photosystem 1. Our first step is our sunlight is absorbed by our chlorophyll. So we have a nice little drawing. We have our chloroplasts. This is our thylakoid stack that we learned yesterday, and our light is coming in to this green little bubble. That sunlight breaks down the water molecule that is right next to it, and our oxygen is released as well as our protons. But this electron from our breaking of this bond from water goes up into the top of photosystem 1. As the electrons move from photosystem 2, I think I just said photosystem 1, our electron goes to the top of photosystem 2. As they move from photosystem 2 all the way to photosystem 1, our electrons, they will pull in a hydrogen. So we are losing energy as we're going all the way down, and that energy is being used to take a hydrogen and pull it to this side of our membrane. If this looks any bit familiar, these are our lipid bilayers. So it's our barriers of our cells, and it's really good for membranes. So it keeps everything on the inside that needs to be on the inside and everything on the outside that needs to be on the outside. But the goal of this part of photosynthesis, this dependent part, is to get as many protons on the inside as we can. So this electron goes all the way down to photosystem 1 and has used its energy to pull in some hydrogen protons. Then we have photosystem 1. See, I can only apologize. It's confusing to have photosystem 1 and 2 be backwards, but they are. So photosystem 1 now has a low energy electron. So our sunlight will hit the electron and excite it and bring it back to the top. This electron is then used to create one of our energy molecules, which is NADPH. So we're charging our battery with this electron that our sun just gave us the energy to do. So now we have a fully charged battery ready to go to our next step. But we also, so that's step 2 is still in photosystem 1. Then this starts what is called the electron transport chain. So what we have next is our electron transport chain. So we have a bunch of this H pluses, which is called a gradient, and they want to move to the other side of our membrane, but they can't do it because our membrane is very, very tight together. So our hydrogens can only go through at one point. But the way our cells work is anytime there's a barrier that wants to be equal. So they would want to have just as many hydrogens on this side as on this side. And that's why it's taken energy to push our electrons into the inside of our membrane, because naturally that's not where they want to go. They want to be even. But this enzyme, we've learned about some enzymes, it's called ATP synthase. So our hydrogen plus molecules can go through our ATP synthase, which will change our ADP into our ATP. So it's really interesting how this works. This part on the bottom, on the top I should say, will actually turn as it's adding our ATP. So we're getting more energy because we're going to be trying to make sugar, which has a lot of bonds and needs a lot of energy to build. So we go through ATP synthase, it turns our ADP into ATP and creates energy. This ATP synthase is a really important and crucial enzyme that I want you guys to remember that name of. But as our ATP synthase turns, we get more energy. So we now have lost our water, but we've created our oxygen and then our energy molecules of NADPH and ATP. So we're ready for the light independent part of our reaction, which is also called the Calvin cycle. So our first step in our Calvin cycle is that we are starting with a 5-carbon molecule. So when I say 5-carbon molecule, that means a molecule that has 5 carbons. Look, and this picture shows 1, 2, 3, 4, 5. It is known as a very, very long name, but abbreviated as RUBP. So this is from our previous cycle of photosynthesis, and it's our starter molecule. So we'll use this starter molecule, and then we'll have to replenish it at the end. But our first step, we'll take our starter molecule and our 6CO2 from our very beginning and combine them. So we're combining a CO2 with one starter to make a 6-carbon molecule. But since we have 6 CO2, we do this actually 6 different times. So all we need to remember though is we're going from 3 carbons in each molecule and we're adding an extra one. So we're making a 6-carbon molecule. This actually is a very, very intensive energy step, and it takes 12 of our ATP and 12 of our NADPH from our light dependent step. So we're using most of our energy by putting this carbon molecule together with this RUBP. Step 2, we actually go from, I think this is actually happening in the same time, we go from our ADP, sorry, let me check my notes just real quickly. I'm so sorry guys, I can only apologize. I had my notes a little bit backwards as I was making this, and after looking through this, I realized I made some mistakes. So our first step is we still start with the starter molecule, and we combine it with a CO2 to make a 6-carbon molecule. Once we do that though, it breaks in, we will break it into 2 3-carbon molecules. Then is our next step, our step 2, and that's when we take in a bunch of 12 ATP and 12 NADPH, and all we're doing is rearranging this 3-carbon molecule into a different 3-carbon molecule. But it's really important that we rearrange it to help create the right orientation, the right geometry for us to start glucose. So our first step is we're fixing these two together and breaking it into smaller chunks, and then we're changing the way that our small 3-carbon molecules look in step 2. Step 3 is we take 2 of these 3-carbon molecules and we make glucose. So we save those for later, but the rest of our 3-carbon molecules have to restart our cycle. So we started with the starter molecule, so we have to reuse it. If we take some sugar out of the cup, or if we take some water out of our botellón, we should make sure that there's enough for the next person. Or whatever analogy you would like to use. That was probably a poor one. But We want to make sure that the next person has enough, but this is the most important part. This 2G3P will get used for glucose. The rest of it will go to restart our cycle. So we're going in, this is just some of the molecules getting taken off, and then the next part is we will restart our cycle. It will take 6 ATP because we got to put some more energy in to get it into the right orientation that we want for our starter molecule. So this cycle works just like any circle, and we just can keep going through it over and over and over as long as we have carbon dioxide and energy. So overall, we are just going back to our table, which shows where everything is and that we can follow it through every step. So we have our light dependent, our light independent, and our overall. We have our water and our sunlight making our energy molecules, our oxygen, and some protons. Our protons get used up, so I should change that. Then we have our ATP and our NADPH plus our carbon dioxide, so our energy plus our carbon dioxide making our glucose. And we finally end with our overall reaction. So just a quick little summary. If this is the level of which I want you guys to understand this very complicated process. So our sunlight enters our chlorophyll and takes the electrons from water. Those electrons move across our membrane, causing a hydrogen H+ gradient. That gradient moves the hydrogens through our ATP synthase, creating a lot of energy in the form of ATP. And then with the help of a lot of that energy, our carbon dioxide is fixed to our other carbon molecules to form glucose. So as long as we understand that we get our energy from our sunlight, that energy is moved through to create different forms of energy that we can use to make the carbon dioxide go into a sugar molecule, into glucose. That is great for now. Another important thing just that I like to mention is, why does photosynthesis even matter? As you can see from the equation, photosynthesis produces oxygen, and we need oxygen to survive. So without plants, we would die. When people suffocate, when people die because they can't breathe, they are dying due to a lack of oxygen, actually, because all of our cells use oxygen, especially our brain. So just some important things. It's without plants in the world, we wouldn't have oxygen, so we wouldn't be able to be around. So just one little thought. I'm sorry about some of those mistakes that we found, but I hope it was clear. If you have questions about some of that, please let me know. Thank you.